I changed my topic a bit. I wanted to talk about yam improvement, but then I thought I should also talk about some other clonally propagated crops. So this is like harnessing the promising fruits of genomics-assisted breeding for improvement of clonally propagated crops. I'm based, as somebody said, I'm in International Institute of Tropical Agriculture, based in a place called Ibadan in Nigeria. Oh, sorry. Okay, so you might be wondering, where is this institute located? This is in Africa, yeah, in Nigeria, which is Africa, very far away from Mexico. It took me 36 hours of traveling to come here. So you can imagine how far it is, right? So we are an institute based here in Nigeria, headquartered. We have about 261 scientists representing 47 countries, and we have a Mexican too. And people should be very proud that he's doing extremely well. His name is Alejandro Ortega Beltran. He's the one who actually encouraged me to come here. And there are 15 countries and 18 locations. We have 6 mandate crops: cassava, yams, banana and plantains, maize, cowpea, soybean. But we also work on other crops like cocoa, coffee, bambara groundnut, and the list goes on. I'm going to talk about yams, cocoa, and coffee, and this is where I work majorly. The campus looks like this. That's the entrance. And this is a 1,000-hectare land area. So this is how the entire, the institute looks like. These are our experimental stations, and we are all, being expats, we are all supposed to stay on campus. So all the 47 nationalities that you see, we all stay on campus. So it's a rich diversity of even human population. And we have a huge lake and a preserved or conserved, rainforest, which represents almost like 400 hectares. So clonally propagated crops, what are they? These are clonally propagated crops which are asexually propagated. Yeah? They are highly heterozygous. They have long breeding cycle. Majority of them takes more than 4 to 5 years, to breed. They have multiple ploidy levels. Majority of the times they have multiple ploidy levels. Poor to no flowering. And even when there is flowering, there is no seed set. Then self-incompatibility. The genetics and genomics of most of these crops are not very well understood. And then there are several pests and diseases. And then, of course, post-harvest losses. And I was mentioning yesterday, there is lack of processing of majority of these crops. Now, what are yams? I don't know if you know yam. However, in US, yams were known to be sweet potatoes. And I kept fighting with them. Come on, sweet potato is Ipomia batatas. And this is Dioscorea. They are completely different species. How can you even link them? But somehow they never understood that sweet potatoes are different from yams. But I think now they know that yams are yams, and these are yams. These are Dioscorea species. There are about 600 species. They are found in Latin America, Asia, Africa, Oceania. They are everywhere. And there are cultivated species as well as, there are only 10 cultivated species, and there are remaining are all wild. In rice, there is only one species. Imagine the amount of work you can do in this crop. There is a huge amount of genetic diversity that is available. And the good thing is, this is source of food and income for smallholder farmers. We work for smallholder farmers. We don't do blue sky research. We do research to have impact on smallholder farmers. And the market value of yam is 13.7 billion USD. Can you imagine that? And that's underutilized. This is a source of carbohydrates for more than and provides 350 calories to more than 3 million people in the world. So that's a huge impact. Okay. So what are the challenges in yam improvement? As I have said in clonally propagated crops, they are all there in this list. But one of the major problems that we have in yams, which makes breeding very difficult, is the flowering. It's a dioecious crop, which means the male and the female should be in separate plants, right? As a dioecious crop. But we also see monoecious. So if you look at this, if you look at this, this is male. I don't know if it's visible. This is female. But occasionally you will see there is this female and this is this male. So, you know, this is crazy. And then you will also find accessions which are not flowering. Then you will also have accessions that will be male this year and female next year. Or female this year and male next year. This is like crazy, right? So that makes breeding really difficult. Very, very difficult. This also means that this crop is under domestication still. It has not been domesticated fully. So we are really now working towards figuring out what is this. Unless we figure out the flowering issues, we cannot develop varieties. Because flowering is what will make us to cross, right? So this is what we are really working on. And as I said, I'm actually a plant breeder or a molecular breeder. So for me, unless I see genetic gain, you know, all these technologies, genomics, phenomics, has no meaning. We have to see genetic gain in farmers' field. And there is a breeder's equation. Genetic gain per year. It depends upon intensity, heritability, and there's something called accuracy. Heritability is the trait that can be transferred from one generation to another generation. If your trait is not highly heritable, you cannot have genetic gain. So this is something that, you know, this is what a breeder is always trying to work on. He's trying to make a gain per cycle of his selection or her selection. So whatever trait you choose for, until you have made an improvement, it has no meaning. So you need to see a genetic gain. This is what we can continuously work on as a breeder. Then we have to also find out which agroecological zones we are working on. You just cannot have a variety for a humid forest which will now work on rainforest. You can't. You need to have a variety that is developed for a certain agroecological zones. So in Africa, you have the Sahelian dryland. That's the Sahel. And that's millet or sorghum belt. This is the humid forest, the West Africa and going up to Central Africa. That's the humid forest. And then you have mid-altitude here in the east. Then you have moist savanna and woodland. So if you see, nowhere you will see yam growing. But yam is integral part of West Africa. So we have to introduce yam in a mixed cropping system, or we have to do it as a monocropping, but we need to develop varieties that will follow both these systems, which makes it much more difficult. Lot of other breeding objectives that you can read here. Now, how do we do this agroecological definition? We need to define the agroecology. If we don't define, then you can't really develop varieties, whether you use genomics or conventional breeding, right? So this is so important. Since we work in West Africa mainly, so what we do is we define the environment where which variety, and that variety should have these characteristics. Once you define the environment, then only you can get all those traits in your variety of interest. are so many traits. How do you define which trait you are going to work on? That's a big question mark, right? And how do you choose that trait? Like for example, I was telling flowering is important, but flowering may not be important because at the end of the day, the yam is already producing the tuber, right? It's producing the tuber and it is actually, people can buy and eat it. So why do I need to work on flowering? I want to work on flowering because I want to develop a new variety, right? And without flowering, I cannot make crosses. However, this may not be an important trait when I go to the farmers. For the farmers, it may be totally different trait. So what we do a participatory and gender sensitive. And what does gender sensitive mean? For a woman, the need for a variety is totally different than a man. A man just wants to have the variety, get it marketed and get the money. That's it. But for the woman, she has to have a variety that can feed her children, that can be marketed, that should have nutrition, that should have blah, 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 blah. The list goes on. So we make sure that we develop varieties which actually is participatory and then gender sensitive. And then we call something trait benchmarking. We put all the traits that are being, rated by the men and the women in different agroecologies, and then we do the trait benchmarking and we select the traits that we should work on. So you remember, genomics is just a technology. So before you apply genomics, you need to do a lot of groundwork. And that should be then you use that technology to develop a variety. So then we do which parents should we use for making the cross, right? We just cannot take any parents and cross it. So once we have done the trait benchmarking, then we go to our germplasm and we try to look at all the parents that we have. And then we try to do the relationship matrix among those parents using something called pedigree. Like you are what you are because of your parents, your grandparents, and your family tree, right? Whatever character you have, it has a benchmark of your family, which is different from your friend sitting beside you. So each of us have a pedigree. Did you understand? And that pedigree gives us our entity or our identity. So we need to do that relationship matrix, we do it. And then once we have selected, see for example, we have selected this particular parent, now we want to see what did this parent already contributed towards development of different varieties. So we look at it and we try to see what is there in that variety, what did it contribute, and if we use it now in our crossing program, is it going to contribute anything more or it is not going to contribute, okay? So there is a lot that you have to do to select a parent. So I will go to a project that was funded by National Science Foundation and Gates Foundation called NSF Bread. We worked on a variety on a crop which is, did I skip anything? Looks like no. So what we do is, in this project we tried to do the chromosome scale whole genome sequencing of water yam. Why water yam? Water yam is globally present. It's also in Mexico, and it's everywhere in the world. Its geographical distribution is everywhere, so we selected this, and that's how the crop looks. It's a vine, so it grows on a vine, so cost of production is very high because you have to stake it, so the farmers, have to spend a lot of money in staking and then mounting and then harvesting. Because you have to, the tubers are here, somewhere down, underground, so there's a lot of work on it. And this project, we had a lot of objectives and a lot of partners that we had. Yeah? So partnership matters. In any project proposal, if you have partners, you will be successful. You want to go alone, it doesn't work anymore in this era. So what did we do is, we first choose the parent that we wanted to sequence. As I said, yam is polyploid, you know, and the bad thing about yam is we still don't know the ploid levels. Like in wheat, we know that it's a hexaploid, right? And we know the genomes, A, B, and B genome. But in case of yam, we don't even know how many genomes are contributed. And we also don't know which plant is, or which accession is triploid, which one is diploid, something like that. So what we did is, we actually did chromosome counting, and then we selected a line which was diploid, right? And then we did that biosequencing, and we also did some RNA sequencing and a lot of things, and we released this chromosome scale, which is already now in public domain. What we did is, we found about 25,000 genes, yeah, which was almost 97.8% complete. And if you see this, you know, we just didn't stop there. We wanted to understand the chromosome structure of this, you know, of water yam. And what we found is that it has a structure like this, Y structure, yeah, and this is called Rabel, R-A-B-L, Rabel conformation. What does this mean? Why did we want to do that? We wanted to do that because we wanted to understand where are the genes located. Because if you don't know where the genes in the chromosomes are located, you cannot make any difference. So what we found is that most of the genes are actually located near the telomeric region, and this is the conformation. So if you really want to make a difference or change, if say for example you say CRISPR-Cas or something like that, it's very important to know the structure of the chromosome. And if you see, we compared this with a lot of other crops, and you see this Dialata, even with Arabidopsis, we have compared. And if you see, our genome is almost complete. No, the gaps are very, very few, while in other crops, the gaps are many more in terms of the genes. We also tried to understand what is the, you know, synteny or the palotetraploidy, like what is the ancestry? What is the ancestor of the current cultivated Dialata? And we found that, you know, there's a lot of conformity with other, you know, other crops within Dioscorea, Trichoppus and Asparagus. What does that mean? That this means that this crop is very ancient. And why then the domestication is still happening? If it is an ancient crop, then domestication should not be happening. But why does it happen? It happens because most of the yams in Africa is in the bush or in the forest. So and they are growing along with other species and wild species. So what the farmers do, or you know, people do in Africa, like in Benin, Ethiopia, and also in Nigeria, they go to the bush. They bring the yam tuber. They bring it, which is not good in shape, not good in length, but they bring it. They will then put it on the soil. They put physical barriers. Okay? And over the years, the morphology of the tuber changes. And it becomes a very nice round-shaped tuber. And they call it as a cultivated species. But now you don't know which cultivated species is it, because there are many cultivated species growing together, and the wild is also growing. So you don't know whether this is wild, you don't know whether it is cultivated, you don't know whether it's an interspecific variety. So there's a lot of complications. So there is, we try to see that. That's why you see a lot of fragmentation in the chromosome, you know, arrangements. So this is what we have found. So what we did is we tried to also understand the medicinal properties of yam. And I'll come there. It's a very important crop. So we also found that, we did the chloroplast and mitochondrial genome analysis, and we compared with a lot of others, and we found that it is very close to one of the medicinal plants that are found in India. Yeah, so this has a very good, high medicinal values, and I'll talk about it a bit later. So we also wanted to, it's just not genome sequencing. What does genome sequencing gives us? We need to do linkage mapping and QTL traits. So what we did is we had 10 crosses with 7 parents, and we wanted to do some QTLs for targeted traits like anthracnose. Anthracnose is a very highly, it's a disease caused by Coledotrichum gliosporoides, and it causes almost 80% to 100% of yield loss. And we also wanted to do some tuber quality traits because of dry matter and other things. So this is how the planting looked like, because we had about 1,500 progenies here, and this looks like a tent, right? As if it's a tent. It grows very fast. And anyways, this is what the crosses were, different crosses. As I said, pedigree matters. And then we had the different traits for those parents. And then then what we did is we tried to look at the relationship between the different parents that were used in these crosses. And what we found is that at some point we lost the pedigree. You see, we can't go back anymore. Here it looks complete, we have the record and we can go back, but somehow we can't make any connection. And this is the problem with breeding. So when in a breeding program, the breeders keep changing, right? Every 5-6 years, a breeder will change to another crop or go somewhere else. So it changes. The records gets lost. That's where digital agriculture is so important. You have to digitize everything. So that's when this got lost, and we could not know, okay, what is the next pedigree? So we could not find, and that's how we could not get some of the traits that we wanted to introgress. So anyhow, we had a linkage map, and for different populations we had, but then we also had something which is consensus linkage map. Because of pedigree loss, we could not use all the populations. So we could use out of 7 only 5 populations, because we then deleted those parents, combinations, because we didn't want to use them, because we didn't know their pedigree as well. So this is, and if you see this picture, it has the same rabble structure. Somehow, you can see that, right? So that's how we had a consensus map. And then we did phenotyping. Let me tell you, genotyping is becoming very cheap. But phenotyping is what is the crisis now. If you don't do phenotyping well, whatever trait association you get is biased. It cannot be a good QTL. So phenomics or phenotyping is the key of this era. And that's why many of the QTLs that has been identified in many of the crops all these years have not been used in marker-assisted breeding, because you just cannot validate those QTLs in another population. So phenotyping is the key. So what we did is we tried to do phenotyping for anthracnose disease, and that's the rating that we used. Generally, this is what is being used everywhere, and that's how it is. And then we also did something in the lab called detached leaf assay. So we brought the leaf. And before that, what we did is we needed to do the genetic diversity of the isolate or Coledotrichum itself. If we don't understand the diversity of the pathogen itself, then how do we actually understand the host-pathogen relationship? So we collected more than 570 isolates from 5 different countries in West Africa, and when we did the genetic diversity, we got the most virulent isolate. So when you test against the most virulent isolate and get a resistant variety, then what happens? then any other isolate it will be resistant, right? So what we did is we got the most virulent one and then we did detached leaf assay. So we detached a leaf, we treated it with the most virulent isolate of Colletotrichum, and then we tried to score this thing. So it was just not in the field phenotyping, but we also did it in the lab. In the field, what happens? In the field, you do not have a control of the isolates. There could be mixed infections. You don't know how many isolates are available. You may do a soil samples analysis today, but tomorrow, again, it may change. The isolate may mutate or something, it may change. So that's why you need to verify it with detached leaf assay. And these are the summary of the phenotyping work. I'll go fast because I have a lot, and the QTLs that we have got. At the moment, we are validating these QTLs. Yeah? And then we did phenotyping for tuber quality, like tuber color. You can see the color difference, you see? Then oxidation, and look at the shape of the tubers, right? Then we did it for oxidation, then dry matter content. Dry matter is very important because in West Africa, they pound the yam. They pound the yam and they make a ball, and that's what they use to eat. They call it swallow. Yeah? So that's what the pounding has to be done. The more the dry matter, the pounding will be better. And then we also found different, QTLs for that. These are the different QTLs which are also being validated by one of my PhD students. So anyway, now this is what I'm coming to, the flowering. Yeah, you see this flowering, you can see it better now. This is the male flower and this is the female flower. And now you will see female here and male here. This is monoecious. And then you have a lot of other combinations of this and no flowering. So now we have found a marker for flowering, which is when it is in heterozygous condition, it will give you a female. And when it is in homozygous condition, it can be a male or a monoecious. So that means what? We still cannot differentiate the monoecious and the male. And we can only do the female. You see, it is so difficult to work on this crop, but it's interesting, right? So then what we now do is we try to do a sex determination at seedling stage. Otherwise, you have to wait for one full, 10 months or something before you can say, oh, whether it has flowered or it's male or female. So what we do is we select only for female. The male and the monoecious, we cannot differentiate yet. But this has helped us a lot. And then I moved to something called germplasm characterization that we always do, and we have done this work already, and using GBS. But just with GBS, after doing this, what we have found is that the breeding lines, the breeding line, you can see that there is not much admixture. The wilds are also not admixture. But when you go to the land races, which is land races, breeding lines, market varieties, when you see, there's a lot of things going on. So in clonally propagated crops, it's common. There's something called synonymy and homonymy. That means different varieties have same name. Like for example, if 4 of you are called with same name, can we differentiate? We can't. Again, there are sometimes different varieties are called with same varieties are called with different names. So there's a confusion. Okay, it has an impact on the seed system. You cannot sell a variety because these kind of issues are continuous, and a market person, what it does is when the tubers come to the market, to just market that tuber, they change the name to a popular name because otherwise nobody will buy it. Only through popular names they will buy, and when somebody comes to buy, they don't know or they don't see the difference in the tubers, right? They just buy it with that popular name. So this goes on a lot in West Africa. And then we use that to develop something like SNP chip. Now we do a regular quality called QA/QC for identifying DNA fingerprinting mainly. We do that, and we do it in a Casper technology using that. So GWAS is something we do. Dr. Philip has explained it very well yesterday, so I don't want to talk more about it. But generally, we also have done some work on GWAS, and we found some traits, but for flowering, senescence, and anthracnose disease. And we all of this, everybody is doing nowadays on network. So I go for something called metabolomics. We also do metabolomics for precision phenotyping. We are doing that. So now what we do is, we use this tuber and we cut it into top, middle, and lower portion. And why do we do it? We do it because we have found that when you cut a tuber from middle, top, and bottom, the germination varies. The top one will germinate first, the middle one will germinate later, and the last one, God knows when it will germinate. So there is a germination difference. That means what? There is accumulation of metabolites in different portions of the tuber itself. So most of the carbohydrates are accumulated on this side, top. And that's how it germinates first, right? So we try to then understand the different metabolites in different portion of the tubers. And this is how, it looks, it's pretty complicated, this thing. But what is that we found a lot of metabolic variation. And there are two species, rotundata and decaensis. They are from West Africa and they always cluster together. And here also we found the same thing. Okay? And what we found is that there's something called tuber dormancy. The tuber, when you harvest it, it does not germinate for 2 to 3 months. So this is dormancy. Potato also has dormancy. So there is a dormancy. So we found a pathway for the dormancy. So we can now break it. We understand now with metabolomics how to break it. And we also did a lot of other studies with other wild relatives to find out some good things. And what I will tell you is, in this particular one, there is a very important component called shikimic acid. Currently, shikimic acid is being done, in a chemical way, unnatural. But Dioscorea has shikimic acid, and the cost is $500 million. Like you can have $500 million. So this is the potential of this crop, underutilized, right? And then we tried to find out what is the difference between land races and improved varieties, and we found a lot of taste and other things. So metabolomics is, actually helping us to understand the crop very well. Now this is what is very close to my heart, right? And thanks to Illumina that I got this award this year, not 2021, I got it in 2022. Yeah, this is a very huge recognition for this crop. And this is allowing me, Illumina used to give $50,000 to earlier awardees. With me, they have increased the award to $300,000. Thank you very much, Illumina. And they made me so popular. They made me so popular. Every day I receive a call for an interview. So I'm so thankful that they have selected, and I'm going to actually genotype or do sequencing of 1,000 accessions not only that, I will do an additional 100 accession or 100 isolates of virus. This is going to make this crop of mine, I call yam as my crop, because when I joined IITA, I had only $3,000 to work on it, while other crops had thousands, 100,000s. I took it as a challenge, and I said, no, I'll work on this crop, and I'll make this crop from underutilized to a most important crop. And today, I think it has got its value, it has got its recognition. Thank you very much again. So I changed my gear to cocoa now. Cocoa, a crop which has probably originated in Mexico, right? One of the origin maybe in Mexico. However, today the highest producer of cocoa is in West Africa. Cote d'Ivoire is the highest producer, and then it's Ghana. And this is how it is, it looks. So you know this is the highest producer. These are the highest producers. But if you see the highest consumer of chocolate is United States. If you ask a farmer of cocoa in West Africa what is a chocolate, they don't know. They don't know that their cocoa can give chocolate. They have never tasted chocolate. They never know that their crop can do that. And how much money do they get? Very little. And how much do you pay for a chocolate? Very high. Right? There is a difference, and there is something called unfairness. Now, 16 of the top 20 consuming countries are Europeans. Sorry, you can read all of that. But what I'm saying is that majority of the cocoa producers are smallholder farmers with just 1 to 3 hectares of land. And then there is something called land tenure. That means if I have 4 siblings, that 1 to 3 hectares of land gets divided into 4. Next year it gets divided again. So what does those farmers have? Nothing, right? It feels. You should feel that. It feels that what are those farmers facing, right? So productivity is not keeping up with the demand. Just because India and China has recognized the value of chocolate and started eating chocolate. Earlier when I was a child, my mother used to say, okay, take a chocolate, and it used to be a candy. So I didn't know that chocolate is chocolate. Now it's only the last 5 years we now started eating. So now that we have started eating, which China and India representing, yesterday you saw 40% of the world's population, America and European countries are now becoming very aware that they will not have their chocolate anymore, right? So now what is happening is demand is increasing very high. There is pest and disease pressure. There is something called cocoa swollen shoot virus disease. This disease is only in West Africa. It's not there in Latin America yet. And that's why there is something called preemptive breeding has to be done so that this disease does not come. Yesterday in many of the presentations you have seen how diseases move from one continent to another continent. And we have a huge partnership with national partners, national institutes, as well as with a lot of private companies called World Cocoa Foundation. And I used to have some funding from World Cocoa Foundation. But the problem with chocolate companies are that they don't want to invest in research. They want to invest on social issues like child labor or something like that. But if you don't invest in research, how can you get, the genome was sequenced I think 25 years back, but still they don't have any trait or QTLs for any trait. For a crop like cocoa which has billions of dollars in it, I don't understand this. Anyways, so we had some money from World Cocoa Foundation where we did DNA fingerprinting. And what we found in our DNA fingerprinting is most of the cultivars that are present in National Institute are all mislabeled. That means what? The parent that you are selecting for making a cross may not be the actual parent. So if you are A and somebody has changed it to B and you think it is A and you take it because you think that this has the traits, but it's actually not A, it's B, your whole process of crossing and breeding is worthless. So this is what we have found, and we made a plea to World Cocoa Foundation that they should make more investment so that we can produce, do some more work and correct those mislabeling. But no, nobody understands and we don't have funding for that. Now, how do you increase the sustainability then you need to have proper environment. You need to have a management. But I also put another arm to this, which is called marketing. You may have a lot of product, but if you don't have a market, then where do you sell your products? So you need to have a market for that. So this is what we say, yeah. Now I'm going to change gear again and go to something called coffee in Nigeria. You have seen the presentation from Dr. Philip on coffee in Ethiopia and others, but this is in Nigeria, and this has only, we had Arabica and many other, in their collection. We wanted to know, is this collection right? Is there mislabeling? So we did some GPS, and we found that as in cocoa, there's also mislabeling in the germplasm maintained in Research Institute. And when we did also morphological characterization, we found that, there are floral characteristics which are completely different. The ones that are, Caniphora, they are actually labeled as Arabica. So there's a lot of, mislabeling. Mislabeling is one of the major issues in any clonally propagated crop, where they are maintaining some gene bank and people keep changing. So we then wanted to know, let us do some metabolomics in it and understand if this is correct. So we did metabolomics, and we found, this is the result from metabolomes, and this is from the SNP. And you can see that actually there was a lot of synchronization. So whatever, mislabeling we found through SNP analysis, we had the same with metabolomics as well. So now all of this boils down to data management. You can do a lot of phenotyping, you can do a lot of genotyping, but if you don't have a system for data management and data analysis, then it's of no use. And that's how the big data is becoming very, very important. So for yams, we have something called YamBase. Again, thanks to Gates Foundation, who has funded us. They gave us $15 million for YAM. So we have done that. But we also developed something in-house, which we call it as YAM Cross. To select the parents, now we can do it digitally. Then we have something, and you people should Google this, seed tracker. We developed it in-house. So now with this seed tracker, what we can do is, we can track where our varieties are, where they have gone. So this is such a fantastic, this is something called certification. It certifies, if you have now developed a variety and you have disseminated to, say, 1,000 farmers, you need to keep a track of it, right? Because that farmer may change. So now everybody gets registered here online, it's on seed tracker, and then we can actually do it. So this is done for cassava, but we have now adopted it for yam, and we are trying to adopt it also for cocoa. Tool which makes digital agriculture or digitization of farmers and the varieties and seed certification really, really better. Now, I have some concluding remarks. I don't have conclusion. Because I cannot conclude. My research goes on, right? My concluding remarks are, what is really required to efficiently and effectively increase the income of smallholder farmers? At the end of the day, if the farmers don't produce anything, we don't have food on our plate, right? So when we do something, we always need to understand that the roots are smallholder farmers, right? So how can we really increase their income, right? The next question is, how can this be done in an environmentally and socially responsible manner? Environment because of climate or whatever, the microbes, the climate, everything, and how is it socially, possible, responsible. We have to be responsible. My next is, a new research culture is required. We cannot do research the way we have been doing. We have been doing research in silos, in silos. I do my research, I publish, I'm done. No, we can't do that way anymore. There has to be a change. We need to come together. All the disciplines need to come together and we need to work together to make a difference. And there is a need for a research and development continuum. Research has to go with development. Then research across disciplines, they have to come together. There has to be innovations. Innovations in such a way that can make a difference in a farmer's field, right? And there are platforms that need to have public and private partners. Public institutes cannot do everything. Private cannot do everything. Everybody has to come together. So the way forward is there has to be a continuous stakeholder engagement. Sorry to the organizers, I am telling you, we needed to have private partners more and more to see what difference can be made. Only the private partners can do that. So another thing that is more important is capacity development. We have to stop the brain drain. Many of us in the developing world, we study in our own countries, but then we go to US or Europe to pursue our career. And that has to stop. We need to do things for our own country. That means the governments of our own country need to recognize the value of us, right? And give us that platform that we can perform. Now, colleagues and collaborators of mine, I can't do anything alone. I have a lot of people who have been working together with me. I have a huge staff and many other students that I have not even put their pictures here. They help me all the time. And then there's acknowledgement to all the funders, because without donors, we can't do anything. And look at the market of yam in West Africa. Look how the yams are stacked and they are being brought to market. This is my last slide. I would like to thank the organizers for bringing me here or to invite me. I'm glad that I came physically. I traveled a long distance, but I'm glad that I came because I can make a one-to-one connection. I'm sure if I would have made this presentation online, the impact wouldn't have been as much as I've been able to do here. Right? Thanks to some of the people, and I have to name them, please bear me for a couple of seconds more. I have to say a big thank you to Dr. Sharma for inviting. I have to thank Dr. Paula for making all the logistical arrangements and to bear with me, you know. And I have to thank Dr. Alfredo, Dr. Victor, and all other, people who have been really a backbone of this first Agrigenomics conference, and I wish you all the luck and best wishes for your next versions. Thank you.